I hope you enjoyed that clip of Alton Brown. See, I told you we'd come back to World War II and the whole carrots and pilots thing. But no, the pilots didn't eat so many carrots that they turned orange. No. That's not to say that you won't turn orange. One of my daughters, when she was very little, had a little friend who loved to drink carrot juice, and this girl's skin was turning orange. Not a danger, it's just a cosmetic thing. All right, so to continue off, I'd now like to switch gears a little bit and turn to some neurophysiology, which I'm really hoping will be review, so I'm not going to spend a super lot of time on this. So we need to know about neurons, which by definition are specialized cells in your body that transmit information. So they're your body's internal communication system. And here is a picture of a very typical kind of a neuron. It's got the cell body, the dendrites, which receive information, the axon, which sends information, and the axon terminals, or the terminal buttons at the end. This neuron is a little bit special because it does have a myelin sheath, a fatty coating, which does speed up the transmission of the nerve signals down the axon. Now, we'll be talking about neurons, but indirectly they're not our primary focus. Instead, we will be looking at a subset of neurons that are a bit more specialized. Those are receptors. So what receptors are a subspecialty of neurons, and their job in general is transduction. That is, to take energy or chemicals from the world around us and convert that information contained in the energy or chemicals into a neural code. So we need to explore a little bit more about neurons and how they work and how they send their information. All things being equal, neurons have a resting potential. That is due to the concentration of ions inside of versus outside of, surrounding the neurons. There is a net difference, and we measure potential differences in units of volts. Turns out there is a very small difference. No surprise, because neurons are very small. We have a potential difference of minus 70 millivolts. This is just when neurons are sitting there not doing anything, pretty boring, but we do get excited when neurons have an action potential. Now they're actually doing something. They are sending messages. As long as the neuron gets enough information, if it gets sufficient input to cause this neuron itself to become activated. When that happens, there is a depolarization phase in which sodium ions enter the neuron. The inside now goes from being net negative to more positive. Well, that's good, except the problem is we need to get things back where they were so this neuron can go back to normal. So there is a repolarization phase in which sodium ions exit the neuron. And now again, it returns back to that resting potential. Words are not the greatest for describing or depicting this. So here is an animation developed by the Department of Biological Sciences. Lots of things going on. Yeah, lots and lots of things. Okay, let me just explain what we're looking at here. We have some sodium ions, some potassium ions. This is the membrane of the cell. Anything below this is inside of the cell. Anything above it is outside of the cell. These weird-looking structures are protein structures in the membrane that will selectively allow certain ions through. Now, with this particular neuron, we are measuring the potential difference in units of millivolts here. You can see we're starting at minus 70, and this graph will show us what happens. So let me reset this. Let's play it again and start it. So let's just watch the ions this time. Okay, what's happening? Nothing. Oh, no, the sodium is entering, and now the potassium is exiting. Okay, now let's watch it again and watch the graph. Okay, and you can see the graph is going up to the peak. Now it's repolarizing, overshoots a little bit, or undershoots rather, and now goes back to its resting state. Okay, so we've got a bunch of moving charged particles, and that is the definition of electricity. Okay, now this is quite a bit of detail. We don't really need to know this for the purposes of this course. there are some things that are going to reappear. So yes, we can measure what's happening with an individual neuron. So this is my kind of hand-drawn version of that graph in the previous slide. And you know what? This actually gives us more detail than we need. This x-axis is time. What I'm going to do is compress this axis in. So we don't have to look at this as a, as kind of a waveform here. We're going to compress it so much that this is what it looks like. A blip, okay? And this one blip is going to be one action potential. And we're going to be seeing a few graphs throughout this course that look like this. Now, the thing about neurons is that they fire spontaneously. We don't completely, totally understand why, but we can measure it nonetheless. In the absence of any stimulus, neurons have a baseline response rate. And that's what I've tried to draw here, tried to draw it to look as random as possible. Now, when there is a stimulus present, a couple of things can happen. The firing rate may increase, so clearly that's an increase over that previous example. This is an example of a decrease. And this is decreased obviously to 0. There's absolutely nothing happening. So again, it's important to think of that. Because of that baseline, there are two kinds of changes, increase or decrease. We can do a thought experiment. Well, what if neurons were quiet? What if they didn't fire during their baseline phase? Well, then there's only one way that they can change, and that would be to increase. So it's kind of a neat way of being able to represent information to a greater extent by having that baseline response rate. Now, this graph on the left doesn't necessarily show the maximum. There are still some gaps, but neurons, of course, do have a maximum firing rate. It's determined by the refractory period. So again, the neuron has to kind of rejigger where it puts all of those ions to make sure the balance is where it is supposed to be. And this takes all of about 1 millisecond. So that is a hard limit on neurons. And that's going to be coming up eventually in the course. So of course, what neurons do is they communicate. So we don't really learn anything by looking at one neuron in isolation. We have to take a look at where they come together, or rather where they almost come together. This, of course, is the synapse, which is technically a little teeny tiny gap between the axon terminal of one neuron, the one that's sending the message, and the dendrite, or perhaps the soma or cell body of another one that's receiving the message. Again, it's a teeny tiny little gap. And if pictures help, okay, here's a picture. So here we have our sending neuron. The impulse goes down the axon, and it is received by this neuron. Now, it looks like they're touching, but it's so close, but there is a teeny tiny gap in here. So how can the neurons communicate if they're not touching? Clearly, this is not an electrical connection. At this point, the nature of the message goes from electrical, moving ions and charged particles, to chemical. So we have these containers called vesicles, and the moving charged particles cause these to migrate to the membrane, and they kind of barf their contents out into the synapse. They go across, and they reach receptor sites on that receiving neuron. Okay, so neurotransmitters are chemical messengers. As far as we understand, it's all about the size and shape. So does the molecule of the neurotransmitter fit into the receptor site? Maybe yes, maybe no. So here too, we can add to our understanding by talking about the threshold. Now, yes, I talked about threshold before, but that was the psychophysical threshold, our experience, the 50% of the time that you can detect a stimulus. Okay, this is the same word, but applied differently. It's now applied to the level of a neuron. And the threshold is how much input does a given neuron require until it, in turn, is activated. And it's not simply a matter of just adding the number of connections, because the connections can go one of two ways. They might be excitatory, which increase the likelihood that the receiving cell will fire, or inhibitory, that will actually decrease the likelihood. Now, the metaphor that's used here with neurotransmitters is a lock and key. So the neurotransmitters are keys. They fit into the receptor sites, which are locks. You take a key and randomly try to stick it into locks here, there, and everywhere. Sometimes it's going to fit. Most of the time, it's not even going to fit. Now, once your key goes into a lock, it can do one of two things. It can either lock it, making it less likely you can open the door, or it can unlock it, making it more likely the door is going to open. I love this metaphor. Okay. Inhibition, we can take a look at. It's due to hyperpolarization. So potassium will exit, or chlorine ions will enter the neuron, and the inside becomes even more negative. Going in the opposite direction, it is not going to have an action potential. Okay, so let's take a look at an example. I'm going to set the threshold equal to +2. We've got 3 neurons, A, B, C, connecting to D, and I want to understand, what does it take to cause cell D to fire? Now, neurons A and B, they're colored green. They have little forked endings, which is schematic. They don't actually look like that, and a +1. C is red, a T-shaped ending, and a -1. Okay, so excitatory and inhibitory. So now I can take a look at every single possibility. What is it going to take to cause D to fire? If A is firing all by itself, that's +1. Is D going to fire? No. B all by itself? No. C, that's a total of -1. No, D is not going to fire. How about A and B simultaneously active? Let's do the math there. 1 + 1 = 2. Yeah, we've reached the threshold. We don't have to go over it. As long as we hit it, that's good enough. If we look at every other possibility, even A, B, and C simultaneously active, that's not enough to cause D to fire. So this is a very simple circuit of 4 neurons, really basic, really simple, but they're actually doing some pretty complicated logical processing, if you like. Now we can try to answer the question of what neurons are doing as a whole. We can understand what an individual neuron is doing and how it connects to another neuron, but on the whole, how do they represent the external world? And this is what our neurons do. There are a few possibilities. The oldest is called specificity coding, and it's kind of the simplest too. And it is based on a one-to-one correspondence between a neuron being activated and a sensation. So if you see red, that's because your red neuron is activated. If you see blue, well, the red has turned off and now the blue is firing. And this can lead to, it was actually kind of a tongue-in-cheek criticism called the grandmother cell argument. Well, if we take this idea and follow it to its logical conclusion, if you look at your grandmother, and you know that you're looking at your grandmother, you can see your grandmother, that's because your grandmother cell is firing. It doesn't matter if you're looking at her from the side or front on or from the top down or whatever, this cell is going to fire, and that's how you know you're looking at your grandmother. Again, this was kind of a tongue-in-cheek criticism of this idea, but the people who proposed this idea were like, yeah, that's kind of what we're talking about. This is not the only possibility. Another possibility is kind of the opposite of this one. It's called distributed coding. So now it's not about one neuron firing. Now it's taking a look at a whole bunch of neurons, and they're not really special, but it's looking at the pattern of activity across a whole bunch of neurons. Now this gives rise to a number of specific possibilities, including temporal coding. So it could be the pattern over time. So maybe how many impulses per second, maybe that is how every different stimulus is getting encoded. Red leads to a higher firing rate and blue is a lot lower, something like that. Something called population coding. This is looking at the firing, not necessarily over time now, but looking at the number of neurons that are firing. So maybe with red, there are 1,000 neurons, and if you see blue, it's only 100 neurons, something like that. And then we can come up with hybrids and much more complex versions like temporal synchronicity. So now it's looking at the firing rates of large numbers of neurons and how that's changing over time. So it's kind of a hybrid of the previous two, getting pretty complex. And then, oh dear, we have another possibility called sparse coding, in which just a small number of neurons, okay, not a single one. Specificity theory would say it's like one neuron. So it takes this from distributed coding. It's a small number of neurons, and it's kind of a cross between specificity and distributed coding. It's a bit more complex. I just want to introduce the concept. We'll see more of this coming up. First, I want to give you an example. So in this example, we're changing a stimulus. We're going to keep it the same frequency and whatever else you want to think of. What we're doing, though, is changing its intensity from 10 decibels to 50 decibels. And I want to use this to illustrate 3 different ideas. For specificity coding, it would say we encode this and we can tell the difference between this because our 10 dB neuron will stop firing and our 50 dB neuron is going to start firing. And that's how we know that, oh, it's gotten louder. According to distributed coding, again, there are a few possibilities. Maybe all of the neurons in some part of your auditory system will increase their firing rate. And maybe in a very nice pattern going from 10 impulses per second to 50. It's probably not going to be that perfect, but let's just go with it. Or maybe the total activity is going to change from 10 active neurons to 50 active neurons. Okay, so there's a few possibilities under distributed coding. And then under sparse coding, well, maybe there's a smaller number of neurons that change their firing rate. Okay, this is all kind of theoretical, hypothetical, but what about the actual evidence for this? Okay, this is pretty difficult to come by. Neurons are teeny tiny. If you want to measure from them, well, organisms tend to be using their neurons and they don't want you poking around in them with like needles and things. But sometimes there are unique opportunities, as happened in 2005. In this study, there were people who were waiting to go for surgery to remedy epilepsy. So epilepsy is a storm of activity in the brain that starts in one area and spreads. If there are no medications that are helpful, people may go for surgery, which will cut out certain parts or disconnect certain parts from other parts, limiting the spread of the storm of activity. So they were trying to determine, okay, where does the storm start and where does it spread to? You can't just kind of go in and do surgery first off. So these people had electrodes implanted in their brains to determine like where is the seizure starting, where is the storm of activity originating. And then they just had to kind of wait around for a seizure to occur. All the while, these electrodes would be monitored. So in these people, the implants were in the medial temporal lobe, a bunch of regions, including the hippocampus and amygdala. And while they're just waiting around, well, you know what? This is a unique opportunity to study what's going on in the brain. You can monitor from, well, about 64 different neurons at once. Pretty neat. So they started this experiment, and they chose to show the participants images of celebrities, a bunch of different photos of the same celebrity, animals, and other objects, and famous buildings. And they wanted to see, are we getting a strong response from one of the 64 neurons if we show, say, the Eiffel Tower, for example? Like, that would be neat. They found one patient who had a neuron that responded pretty significantly to a bunch of pictures of Jennifer Aniston. It wasn't so crazy about Braniston, so Brad Pitt and Jennifer Aniston together. So their neurons got pretty excited, or one neuron got pretty excited by Jennifer Aniston. Here are some of the pictures that they showed, and you can see the response profile from some of the neurons that they were monitoring. So there's a picture of Jennifer. There's another picture. There's another picture. Another. Oh, that's a nice picture. That's a very nice picture. That's a picture. Here's Brad. Brad. You can see the neuron is not so excited by that. The neuron is very excited by Jennifer. I have neurons that are excited by Jennifer Aniston too. Here's some pictures of some athletes, some other women actresses. Leaning Tower of Pisa, there's a spider and a snake, and if you take a look at the response profile, it's not really the same. These ones really stand out. So it looks like they've latched into a neuron that is all about Jennifer Aniston. That's pretty lucky. In another participant, their neuron was very strongly excited by Halle Berry, by some photos of Halle Berry, a drawing of Halle Berry, Halle Berry dressed as Catwoman from that abysmal Catwoman movie. And look, even the name Halle Berry, look at that response profile. Now that looks very different from this photo of Halle Berry, but you get a very, very strong response. That's amazing. What does this tell us? Okay, it is evidence for invariant. the neurons don't change their preferences. The Jennifer Aniston neurons today are going to be Jennifer Aniston neurons tomorrow. They are sparse, that is, they are selective, not just responding to pictures of female faces, and also explicit. So, strong firing to Jennifer Aniston, really no firing to the Leaning Tower of Pisa. Now, this was very exciting work when it came out, and it led to some follow-ups. They did another follow-up in which they found multisensory neurons in the medial temporal lobe. For example, in this person, here are neurons that respond to photos, letter strings, and even hearing the name Oprah Winfrey spoken out loud. So this is a neuron that is stimulated by photos of a person, seeing their name, and hearing their name spoken, which is amazing. And you know what? This is a whole lot sexier than a grandmother cell. Are these grandmother cells? Have they actually found them? No, that's not universally accepted. Now, if you go back and look at the response profile to Jennifer Aniston versus other things, it's not that the neurons are silent. If they're looking at the Eiffel Tower, for example, they had to come up with a cutoff of, we'll say, this is a strong response, and then anything below this is not a strong response. So it's not absolutely exclusive. Do you think that they had the amazing good fortune out of the billions of neurons to tap into the one single Halle Berry neuron? What's the likelihood of that? And then also in the same study, find a Jennifer Aniston neuron in another person. What are the odds? The odds are against you. Let's think about where the electrodes were. The medial temporal lobe, that's involved in emotion, which may moderate neural activity. Is this all about visual perception? Not necessarily. We can't conclude that definitively. It's possible that you like Jennifer Aniston, whether you know that or not, and you might get a bit of an emotional response, and maybe that's what's being measured. It's not the visual identification of Jennifer Aniston. And what is definitely the case, these are not the storage areas. This is not where our memories are stored, in the medial temporal lobes. No, it is involved in the storage of memories, but it is not where they are actually stored, which is something that we were actually looking for when we're looking for these elusive grandmother cells. So I'll be exploring this idea of sensory coding as we move forward. It is extremely difficult and tricky. There isn't a lot of research on it, so unfortunately I can't tell you exactly what the sensory coding for all of our sense modalities is, but it's something to keep in mind. All right, have a look at the E-class lecture quiz based on this topic.